Let's talk about something we all think we understand: death. What if it's not an off switch? What if, for hours after you're gone, your brain is actually buzzing with a secret, organized life of its own? Today, we're diving into a scientific detective story that could redefine the very line between life and death.
Think about it for a second. It's a pretty wild idea, right? We imagine death is instant, a flip of a switch. But what if it's more like a controlled shutdown, a biological program that keeps running long after the lights go out?
So this whole thing kicked off with a huge puzzle. Scientists noticed something really strange. When they looked at the genetic activity in a fresh, living brain, it looked totally different from a brain studied after death. The researchers called it an entirely unique pattern, which is basically science speak for, whoa, what is going on here?
And that discovery created what we're calling the post-mortem puzzle. You see, if the brain tissue we use for all our research is so different from living tissue, then what have we actually been studying all this time?
And this isn't just some academic thought experiment. This is a huge deal. Most of what we know, or think we know, about diseases like Alzheimer's or schizophrenia or autism comes from studying brains after death. So if that tissue is fundamentally changed by the very process of dying, it raises a massive question. Could we have been mistaking the biology of death for the signs of disease? To solve this puzzle, the scientists did something that was just brilliant. Instead of just comparing a before picture and an after picture, they decided to watch the whole movie. They set up an experiment to essentially create a time-lapse of a brain dying.
Okay, so there's one key term here that you need to know: the post-mortem interval, or PMI. It's really simple. It's just the amount of time that's passed since someone has died. And the researchers wanted to know what exactly happens to the brain's genes as that interval gets longer and longer.
And the experiment they came up with, so simple, it's genius. They got a piece of fresh, living brain tissue. This was tissue that had to be removed during a surgery. Then they just left it at room temperature to perfectly mimic what happens after death. And over the next 24 hours, they took samples to watch, step by step, what changed. And what they saw was just wild. Let's walk through it. At hour 0, you got a normal living brain. But just 4 hours in, the thinking cells, our neurons, are already starting to check out. But at the same time, another type of cell, the glia cells, are just getting warmed up. By 12 hours, the glia are at their absolute peak. And by 24 hours, the whole scene has changed. The thinking genes are practically gone, but those glial genes are still going strong. You know, the researchers themselves put it perfectly. From the point of view of the brain cells, being suddenly removed from a living person is nothing short of a catastrophic event. And their clever experiment gave them a front-row seat to see the brain's emergency response protocol kick in. So what is actually happening in there? Well, it all boils down to a tale of two very different kinds of brain cells. And here is the single most important finding. This is not random decay. The brain isn't just passively falling apart like a piece of fruit rotting on the counter. No, it's an active, highly organized process, a coordinated reciprocal shutdown. Okay, let's look at the evidence. First, what doesn't change? These are the housekeeping genes, the ones that basically keep the basic machinery of the cells running. And look at that. Their activity is rock solid. This is our first huge clue that this is not random decay at all. The cells are still working. But now, look at the neuronal genes. These are the thinking genes, right? The ones behind our memories, our personality, who we are. The chart tells a pretty dramatic story. Their activity just plummets. This is the part of the brain that makes us, us. And it's going offline, and it's going offline fast.
And now, for the big twist, the real shocker. What about those other cells, the glial cells, the brain support staff, the cleanup crew? Well, they don't fade away. They don't even stay the same. Their activity goes through the roof. They literally get bigger, sprouting these long arm-like appendages. They roar to life after death, which, yeah, is why some people have started calling them zombie genes.
So that's the whole story in a nutshell. It's this beautiful inverse relationship, a coordinated handoff. As the thinking neurons power down, the cleanup glia power up. So, okay, this is an incredible biological story. But why does it really matter? Well, this discovery has the potential to rewrite decades of brain research.
I mean, just look at this list. Schizophrenia, Parkinson's, autism, Alzheimer's. The hunt for cures for almost all of these devastating disorders relies heavily on post-mortem brain tissue. And now, all of that research, every paper, has to be re-examined through this brand new lens.
And here is the core problem, the real danger. Imagine you're a scientist studying an Alzheimer's brain. You see all this glial cell activity, and you think, aha, this must be a hallmark of the disease. But what if it isn't? What if what you're seeing is just the brain's natural healthy shutdown sequence? It means we might have been chasing the wrong clues for years.
All of this leaves us with one final, profound thought. We always thought of death as an event, an endpoint. But this research shows it's a process. It's an organized biological program. And if a brain that we consider clinically dead is actually this busy, with genes firing up and cells physically growing for hours, it just makes you wonder, what other biological secrets does that final chapter of life really hold?